This is part 2 in the series of videos on how to use Shape Genie. And in this video, I will teach you how to calculate an index or multiple indices based on the output data from Shape Genie. So this is the CSV file called output, which I've opened in Microsoft Excel. And first thing I want to point out here, notice this message up here in yellow, possible data loss. That is because you've opened a CSV file within Excel. And if you do any calculations, plot any graphs, or change any colors, if you don't save the file as an Excel file, you will lose all of that work, okay? Because CSV files are only numbers and commas. Okay, so it's very important to, at the beginning, to start with, before you do any calculations, is best practice to click Save As or File Save As. And then you can click right here on the file format option and click on Excel workbook to save as, right? And you can add a name here. You can say Excel, okay, and click save. Make sure you pick this option, Excel workbook. And once you do that, that message appears because now this file is saved in Excel format. So it will store all of the equations and calculations and graphs that you add. So let's now explore this data. So there are 3 samples here. Each column is a sample. And the first row is a header row that contains column names. If you click on the number 1 here on the left, and you click on the Home tab, and then Wrap Text, you can see the names more clearly. And here we know from part 1 that I uploaded images called heart-1, heart-2, and heart-3. All these other words attached afterwards are due to the processing pipeline. It attaches new suffixes at every step, but the core name is still at the beginning. So each sample is its own column. And if you scroll down, they all end at, they all have 71 rows, but they end in Excel at row 72 because, again, the first row is a header row. And they all have the same number of rows because in this case, these three images, we chose to analyze them through the radial grid method. And in the radial grid, typically there are 71 rows. But if you analyze your shape through the parallel grid, then each column or each sample might have a different number of rows, depending on how tall the object is, or the shape is. So just something to know about how many rows to expect in the radial grid system versus the parallel grid system. So this each row is a frequency, right? So let's plot the first sample to see how it looks. So I'm going to click on column A and then hold shift and click on column B. I select both. Okay, I click up here so that it selects everything in these 2 columns. Okay, that's the point of clicking on these capital letters A, B, C, D up here. So I've highlighted A and B by, again, I click on A, I hold the shift key and I click on B. Now both are highlighted. I go to insert. In terms of recommended charts, I just click on that and I'll pick this individual bar chart. And so there it is. Okay, so this plots, okay, each of these from rows 1 all the way to 71. And you see this is the frequency spectrum representing the heart shape called heart-1, which we uploaded in part 1 of this series of videos. And if I wanted to take a look at these smaller bars more closely, then what I can do is I can double click on this y-axis here, and then I can go to these axis options. Sometimes if you don't see it, you have to click on text options and then back to axis options, and then you'll see this drop down menu. And I go to bounds right here, maximum, and I'll pick changes from 40 to 5,000, right? And I hit return and look at what happens to this graph, right? The y-axis has been truncated. So now I can appreciate the differences in the smaller bins more. Okay, and of course the first bin, which is very tall, right, it gets cut off, right, at 5,000 because it was much larger. In fact, it was 33,000. Okay, 848, as we can tell from cell B2. All right, so that's if you want to graph Excel, graph one sample. And here, just a quick Excel tutorial. If you want to add these two samples, you can click here, this blue square, okay, and pull it over, and look what it does. It adds, okay, the two other samples to your graph. And then you can undo that by pulling it back like this, okay? And a neat thing about Excel is that I can click on this graph, click copy, Control C, click somewhere else outside of the graph, and then I click Control V to paste it, and now I have the identical graph, okay? But what I'm going to do now is I'm going to click on the edge of this blue and drag it over one time, okay? And now it's graphing, okay, column C. So this first graph, if I double click it, I see it's column B. If I double click on this one, it's column C, and it copied the name of column C. Here it says heart 2, up here it says heart 1. You see that? So, and you can do the same, of course, if I copy. Okay, let's just start with this one. Copy this, click here, paste, okay, and then I'm going to double click on this graph, and I click on the edge of the blue and I drag it over to heart 3, and now I plotted each of the three individually. You see that? And you can modify the axes in each of the three as we did before. All right, so that's how to graph each of these frequency plots. So now let's talk about how to calculate an index, right? Because each of these samples, right, has 71 values, and that's multi-dimensional and complicated for the human mind to assess. And so we want to derive scalar values, that is single numbers, to represent each shape. The simplest one, let's go to the bottom, is to calculate an index called sum all bins, right? So we add up all bins in all rows to get one number. And to do that in Excel, I click sum, or excuse me, I click equal, SUM for sum. I click open parentheses. I go to the top, I click where I want to start, and I drag down. I let go, I can scroll up, and then I can extend this dotted box by clicking on the bottom right hand corner, dragging down. I let go, I scroll up, and then again I go for the bottom right hand corner, and I drag all the way to here. And once I'm done, I hit return, and here's the answer: 56,410.2776. Right, to check if I highlight the right range, I can click on this cell and see up here the equation is sum, open parentheses, B2 to B72, which is correct, because this cell, it starts at row B2 and it ends at column B, okay, row 72. So it's B2 to B72. And a visual way to check the range that you selected is to double click on the cell, and you can see a blue box appears. And when you scroll up, you can see where the blue box begins and where the blue box ends, right? So that's a visual way of checking what range you are calculating on. Right, to get out of editing mode, press escape. There you go. If you're in editing mode, after you double click and you accidentally type something in, okay, that's going to cause an error, and it's okay if you type, multiple things in, right? To get out of editing and to undo all your typing, just press escape, and it reverts back to the original equation. Okay, so again, if you're in editing mode and you accidentally type something, which happens, just press escape to go back. All right, okay. So this is the index called sum all bins, because that's what we did. We summed all bins, okay, for heart 1 in column B. Press escape to get out of editing mode. To do the same for the two other samples in columns C and D, what I can do is I click on this cell, which I know is an equation, and I find the bottom right hand corner, there's a green square. I click and I drag it over. I let go, voila. It has calculated the same equation, but for each of the corresponding columns. You see that? And I'm going to press escape. So this is an index called sum all bins, and we've calculated it for each of our three samples. Okay, and if we wanted to graph it, we could. Right, I can highlight this. I go to insert, click on recommended charts, and here's a chart a bar chart. See that? It shows the values of this index for each of the three. Okay. All right, so let me just delete that. Okay, so that's a common index to calculate. Sum all bins is usually the first one we do, but it's not necessarily the best one. Other common ones, right, I will show you now. So first, most of the difference, or a lot of the difference, can be found in the first 10 bins, typically, right? And so, I will show you that in this tutorial video. So first, since this is very long in terms of many rows, I'm going to click on, row 12 here, highlight whole row. Then I'm going to click on View tab, and I'm going to click on Freeze Panes. And what that does is now when I scroll, right, the rows 1 to 10, or it actually is rows 1 to 11 on the Excel sheet, always are visible, right, even if I scroll up and down. Okay, so this way I can do calculations here and show you exactly where the range is up here. Okay, so for example, right, say I want to calculate an index called bin 2 divided by bin 3. Okay, and so what I do is I click this cell, all right, and I click press equal, and then I go up here and I click on bin 2. Okay, or actually this is bin 2. Okay, though its address is B3, and I press the / for divide, and then I click on bin 3. It's in red. See that? So B3 over B4, okay, is 2 over 3. If you look at the row names here, 2 over 3. Okay, in the Excel address, right, it's B3 over B4. So I hit return, and that's the ratio, right? It's 0.7. So if I double click this, it shows me the range, right? And it shows me the color, blue over red, right? Blue over red. Blue is here bin order 2, right? And red is bin order 3. Okay, so I'll press escape to get out of editing mode, right? And so I want to repeat this same calculation for the two other samples. So I find this bottom right hand corner, I drag and let go, and there it is. This is the ratio, okay, of an index called bin 2 over bin 3. All right, and you can do the same for bin 3 over bin 2, right? It's very similar. So I press equal, or first I click on the cell where I want to calculate, and I press equal, and then here I find bin 3, which is here, and I click that, and then I press / and then I find bin 2, I click it, and now hit return. Okay, and so I double click on this equation, it tells me, all right, this is what I am calculating. This is the range here, right? Blue over red, which is what I want, 3 over 2. So I'm gonna press escape to get out, and I'm gonna click this bottom right hand corner, drag to the right, let go, and now I've calculated the same thing, okay, for all 3 samples. Okay, now why would I do bin 2 over bin 3 and then bin 3 over bin 2? It's because you should do both. These are common indices that show differences often when we compare various types of shapes, and one may reveal results while the other one does not. So I recommend trying both. Okay, so another common index we have to calculate is summing bins 2 to 7. All right, so here I'm just going to widen this a bit. Okay, so for here I'm going to click equals sum open parentheses. I'll go up here, I go from 2 down to 7. Okay, I let go. You see that this is 2 to 7 right here. I hit return, and then I want to replicate this across these samples. There you go. Okay, and then another common index that we find to be useful is sum bins 3 to 7 as opposed to 2 to 7. Okay, I press equals sum. Okay, 3 down to 7. And here when I say 3 to 7, I'm talking about the bin order, right, which is this first column here, not the row number on Excel. Okay, this is the bin order on the first column. So I'm going to hit return, and then I'm going to double click. Yep, that's right. It's 3 to 7. Okay, I press escape, and then I'm going to drag the bottom right hand corner, let go, and now I'll calculate the same index for the other columns. Okay, so you can graph each of these as bar graphs if you want. Okay, And so here's something very important. So I could have calculated each of these three up here, right? It doesn't matter because when I press in the equation here, right, it tells me to search for, right, which range I want to calculate. So it doesn't matter where I place these calculations, okay? I just place it down here because when you have many, many hundreds of samples, say, here we only have 3, right? It's often useful to sort them, okay, all these samples, right, from high to low for one of these indices, right? And so when you put it down here, all your indices underneath these, the last row, then you can sort all your columns, right, from high to low according to one of these rows. It just makes it easier to analyze. Okay, so that's it. So that's how to calculate indices based on the CSV output, right? And then you can graph the indices, okay? So here I'm just going to do something to show you. It makes your graphing easier. Okay, I'm going to right click on this row here and I'm going to insert, okay, one row above. So what I'm going to do is I'm going to, all right, type in part 1, part 2, part 3. You can copy and paste from above if you want, but I don't want to copy the full name. I just want the first few characters. And so now when I highlight this and I go insert, okay, I choose, okay, recommended graphs, and I click a bar graph. Right now it has the names of each of these headers down here. See that? It makes graphing easier. And if I want, I can click and drag this down here, and now it plots this index, bin 2 or bin 3. Right? Notice for bin 2 or bin 3, part 2 is the smallest of the 3. But if you go to bin 3 or bin 2, it's now the largest. Okay? Again, both indices work, but you have to try both to see which one gives you the cleanest results, especially if your goal is to develop a machine learning model based on these numbers to make predictions on unknown samples. You want an index that gives you the clearest separation between your groups. Right, so I'm going to drag this down here just to show you that we can sum or graph this index called sum bins 2 to 7. Okay, and the graph picks up on the name of your index right here in red. Okay, and in purple is the name of each of your samples. So just a little tidbit on making your graphing easier by inserting a row up here and then adding in your sample names. I would highly recommend visiting our website, brainscannology.com, and then clicking on this tab called Use Cases. In this use case page, we have many examples of how we measure different shapes and the different indices that we calculated. Right? There is no one best type of index. It's all you have to explore in your CSV output file. And you can watch each of these videos as we've applied it to different types of images that we've analyzed, whether it's hand-drawn images like these circles, these spirals, or the shape of fingers. Here, the shape of hands. Here we have the shape of crescent cells. Here we have the shape of cells in 2D culture. Here are shapes of 3D organoids, okay, that we've imaged in 2 dimensions. Here is a video on measuring the lesion in shoulders called Hill-Sachs lesions. This is a tutorial or a use case video on the shape of lesions in the vertebral units in the human spine. And here are use case videos on material science, right, and electronics. Right, this is the shape of crystals within a metal, right, and here is the shape of liquid droplets. So Shape Genie has many uses, right, even outside of life sciences. Here we're measuring the shape of these rocks, okay, this traditional scale of rocks that is taught in geology, and we can show that we can measure precisely each of these shapes within this iconic scale. Right, so watch each of these videos to learn about how we calculated indices in each of these cases so that you can get an idea of how to best explore your data that you get from Shape Genie.